So in this video, we're going to look at the chi-squared test, and we're going to look at when we use it and how we interpret it. So the chi-squared test is done to compare observed and expected. And the really important thing with the chi-squared is that you deal with frequencies, which in other words is a count. You're counting something. So let's look at some examples of where it's used. So a chi-squared test is used to determine if there is a significant difference between two sets of data, but this data must be frequencies. So for example, if I'm comparing, if the number of sixth form boys that wear glasses is significantly different to the number of sixth form girls, I would expect that it should be even, and I'm going to compare whether our actual values are significantly different to our expected values. So we have a null hypothesis. Our null, if you remember, is no, if you've watched the t-test video. So we would say there should be no difference between the number of sixth form boys that wear glasses and the number of sixth form girls. And we're trying to do a chi-squared test to see whether that is true. So, for example, you're looking at the distribution of snails on different species of seaweed on a beach. So my null hypothesis is there is no statistical difference in the distribution of the snails on the different species of seaweed. So it simply is, like we said before, the no bit. My results look as follows. So if you look at it, there are certainly differences. So there's only 5 spiral rack and 2 other algae, but 10 egg rack, 38 bladder rack, and 45 serrated rack. So looking at it and looking just at the raw data, there does look like there's a difference. It looks like the animals prefer the serrated and bladder rack to the egg rack, spiral rack, or other algae. The question is it significantly different? So if the null hypothesis is correct, that there is no difference, then we would expect the 100 snails to be distributed evenly. So our expected value should be 20 for all of them. All we've done is take that 100 and say we'd expect them to be evenly distributed along the 5 different types of seaweed, which gives us 20. So at first glance, there does look like there is a difference. 45 is bigger than 20, 38 is bigger than 20, 10 is less than 20, 5 and 2 are less than 20. The question is it a significant difference? So we know it's different, but is it enough to say that the difference is not due to chance? That's what you're asking yourself in the chi-squared test. Is the difference between what you see and what you expect big enough to say that the difference is not due to chance? And to do that, we have to do a chi-squared test. So you do not have to calculate the chi-squared test in your exam. Same for the t-test, same for the Spearman rank. But you do have to be able to understand it. You may be asked to calculate expected values, especially when dealing with genetic crosses, but possibly other things. And you do need to be able to how to calculate the degrees of freedom. So degrees of freedom is the number of categories minus 1. So if we go back, we have 5 categories here, 1, 2, 3, 4, and 5. And therefore, we have 4 degrees of freedom. We always look at the 0.05 level for our critical value. Remember, we said that the 0.05 level is the 5% probability. We want to be able to say where there is less than 5% probability that the difference is due to chance, or more than 5% probability that the difference is due to chance. So here is our critical value table. We go down the 0.05 level, across at the degrees of freedom 4, and we get a critical value of 9.488. Now remember, that is the minimum value that your chi-squared must be in order to say that there's less than 5% probability that the difference is due to chance. Okay, so that is the minimum value we need to get to be sure that it is not just due to chance. So if we go back to our value, if our value is bigger than the critical value, so remember our critical value was 9.488. So if we get a value like 10.212, for example, that's bigger than our critical value, we can say that our difference is significant, Note differences in capitals. That's to highlight that it should be the word difference, not results. We're looking at the difference. We can reject our null hypothesis. So there is less than 5% probability that the difference is due to chance. And that can also be written as P is less than 0.05. That's another way that can be written in the exam. It means the same thing. Okay, so for example, scientists studied 1,500 births. They recorded the mass at birth of each baby and the nature of the Chi-R gene in the mother's genome. Some of their results are shown in the table. So we're dealing with number, okay, and we're looking at whether something is present or absent. The scientists used a statistical test to test the following null hypothesis. The presence of the KIR2DS1 in the mother's genome does not affect the frequency of births. So that's the null. Now, the question is which statistical test? Well, if we're dealing with number, we're dealing with chi-squared. This is our observed. We would compare it against our expected, which should be evenly split in theory. They're not going to ask us to do it. They're just going to tell us about p-value. So the scientists calculated a p-value of 0.03. What can you conclude from this result? Explain your answer. So first thing you need to ask yourself, we don't have any critical values here. They've told us that p equals 0.03. Now, if we go back to the previous slide for a minute, you'll see that we said that if p is less than 0.05, we know our difference is significant and we reject our null hypothesis. So in this particular case, p is equal to 0.03, which is basically less than 0.05. So therefore, our conclusion is going to be that we can reject our null and that there is less than a 5% probability that the difference is due to chance. Okay, so we're just applying what we learned before. The idea that we need to look for is the value that we're given, which is here, is that more or less than 0.05. If it had been more than 0.05, so let's imagine that the p-value, let's imagine that they told me in the question that they did the calculation, they did the statistical test, and p was equal to 0.08. So if that was the case instead, which it isn't, then that would mean p was greater than 0.05. And I would say that there is more than 5% probability that the difference is due to chance. And I would accept my null hypothesis. But that wasn't the case here. So I'm going to rub that out. Okay, the case was that p was 0.03, which is less than 0.05. So that is how you apply the chi-squared test. Go and practice some questions on it.